Hello, my amazing tribe. I'm excited to connect with you through this video. Welcome, everyone, to our latest video where we'll be exploring placenta. The placenta, placentas or placentae, is a temporary embryonic and later fetal organ that begins developing from the blastocyst shortly after implantation. It plays critical roles in facilitating nutrient, gas and waste exchange between the physically separate maternal and fetal circulations, and is an important endocrine organ, producing hormones that regulate both maternal and fetal physiology during pregnancy. The placenta connects to the fetus via the umbilical cord, and on the opposite aspect to the maternal uterus in a species-dependent manner. In humans, a thin layer of maternal decidual endometrial tissue comes away with the placenta when it is expelled from the uterus following birth, sometimes incorrectly referred to as the maternal part of the placenta. Placentas are a defining characteristic of placental mammals, but are also found in marsupials and some non-mammals with varying levels of development. Mammalian placentas probably first evolved about 150 million to 200 million years ago. The protein syncytin, found in the outer barrier of the placenta, the syncytia trophoblast between mother and fetus, has a certain RNA signature in its genome that has led to the hypothesis that it originated from an ancient retrovirus, essentially a virus that helped pave the transition from egg-laying to live birth. The word placenta comes from the Latin word for a type of cake, from Greek placenta placenta, accusative of placis plakos, flat, slab-like, with reference to its round, flat appearance in humans. The classical plural is placentae, but the form placentas is more common in modern English. In the following segment, we'll be examining evolution and phylogenetic diversity in greater detail. The placenta has evolved independently multiple times, probably starting in fish, where it originated multiple times, including the genus Poeciliopsis. Placentation has also evolved in some reptiles. The mammalian placenta evolved more than 100 million years ago and was a critical factor in the explosive diversification of placental mammals. Although all mammalian placentas have the same functions, there are important differences in structure and function in different groups of mammals. For example, human, bovine, equine, and canine placentas are very different at both the gross and the microscopic levels. Placentas of these species also differ in their ability to provide maternal immunoglobulins to the fetus. In this segment, we'll be unraveling the complexities of structure and exploring its multifaceted nature. Placental mammals, including humans, have a chorioallantoic placenta that forms from the chorion and allantois. In humans, the placenta averages 22.9 inch in length and 1 inch in thickness, with the center being the thickest and the edges being the thinnest. It typically weighs approximately 500 grams, just over 1 pound. It has a dark reddish-blue or crimson color. It connects to the fetus by an umbilical cord of approximately 1 inch in length, which contains 2 umbilical arteries and 1 umbilical vein. The umbilical cord inserts into the chorionic plate, has an eccentric attachment. Vessels branch out over the surface of the placenta and further divide to form a network covered by a thin layer of cells. This results in the formation of villus tree structures. On the maternal side, these villus tree structures are grouped into lobules called cotyledons. In humans, the placenta usually has a disc shape, but size varies vastly between different mammalian species. The placenta occasionally takes a form in which it comprises several distinct parts connected by blood vessels. The parts, called lobes, may number 2, 3, 4, or more. Such placentas are described as bilobe, bilobipartite, trilobe, trilobitripartite, and so on. If there is a clearly discernible main lobe and auxiliary lobe, the latter is called a subcentriate placenta. Sometimes the blood vessels connecting the lobes get in the way of fetal presentation during labor, which is called vasa previa. As we enter this new chapter, let's navigate the complexities of gene and protein expression and unravel its multifaceted nature. About 20,000 protein-coding genes are expressed in human cells, and 70% of these genes are expressed in the normal mature placenta. Some 350 of these genes are more specifically expressed in the placenta, and fewer than 100 genes are highly placenta-specific. The corresponding specific proteins are mainly expressed in trophoblasts and have functions related to pregnancy. Examples of proteins with elevated expression in placenta compared to other organs and tissues are PEG and the cancer testis antigen PAGE, and expressed in cytotrophoblasts, and KISS expressed in syncytial trophoblasts, and PAPA and P expressed in extravillous trophoblasts. Now, let's shift our attention to development. The placenta begins to develop upon implantation of the blastocyst into the maternal endometrium very early on in pregnancy at about week 4. The outer layer of the late blastocyst is formed of trophoblasts, cells that form the outer layer of the placenta. This outer layer is divided into 2 further layers, the underlying cytotrophoblast layer and the overlying syncytial trophoblast layer. The syncytial trophoblast is a multinucleated continuous cell layer that covers the surface of the placenta. It forms as a result of differentiation and fusion of the underlying cytotrophoblasts, a process that continues throughout placental development. The syncytial trophoblast contributes to the barrier function of the placenta. The placenta grows throughout pregnancy. Development of the maternal blood supply to the placenta is complete by the end of the first trimester of pregnancy, week 14 DM. As we progress through this video, let's shift our attention towards maternal placental circulation and uncover its hidden depths. In preparation for implantation of the blastocyst, the endometrium undergoes decidualization. Spiral arteries in the decidual are remodeled so that they become less convoluted and their diameter is increased. The increased diameter and straighter flow path both act to increase maternal blood flow to the placenta. There is relatively high pressure as the maternal blood fills into the loose space through these spiral arteries, which bathe the fetal villi in blood, allowing an exchange of gases to take place. In humans and other hemicurial placentals, the maternal blood comes into direct contact with the fetal chorion, though no fluid is exchanged. As the pressure decreases between pulses, the deoxygenated blood flows back through the endometrial veins. Maternal blood flow begins between days and is approximately min at term. Now, let's shift our perspective and explore fetoplacental circulation through a fresh lens, unlocking new perspectives. Deoxygenated fetal blood passes through umbilical arteries to the placenta. At the junction of umbilical cord and placenta, the umbilical arteries branch radially to form chorionic arteries. Chorionic arteries, in turn, branch into cotyledon arteries. In the villi, these vessels eventually branch to form an extensive arteriocapillary venous system, bringing the fetal blood extremely close to the maternal blood, but no intermingling of fetal and maternal blood occurs. Placental barrier. Endothelin and prostanoids cause vasoconstriction in placental arteries, while nitric oxide causes vasodilation. On the other hand, there is no neurovascular regulation, and catecholamines have only little effect. The fetoplacental circulation is vulnerable to persistent hypoxia or intermittent hypoxia and ruxygenation, which can lead to generation of excessive free radicals. This may contribute to pre-eclampsia and other pregnancy complications. It is proposed that melatonin plays a role as an antioxidant in the placenta. This begins at day. Let's now venture into the realm of birth and explore the fascinating intricacies it holds. Placental expulsion begins as a physiological separation from the wall of the uterus. The period from just after the child is born until just after the placenta is expelled is called the 3rd stage of labor. The placenta is usually expelled within minutes of birth. Placental expulsion can be managed actively, for example by giving oxytocin via intramuscular injection followed by cord traction to assist in delivering the placenta. Alternatively, it can be managed expectantly, allowing the placenta to be expelled without medical assistance. Blood loss and the risk of postpartum bleeding may be reduced in women offered active management of the 3rd stage of labor. However, there may be adverse effects and more research is necessary. The habit is to cut the cord immediately after birth, but it is theorized that there is no medical reason to do this. On the contrary, it is theorized that not cutting the cord helps the baby in its adaptation to extrauterine life, especially in preterm infants. Get ready for an enlightening exploration as we dig into microbiome and understand its role in the broader context. The placenta is traditionally thought to be sterile, but recent research suggests that a resident, non-pathogenic, and diverse population of microorganisms may be present in healthy tissue. However, whether these microbes exist or are clinically important is highly controversial and is the subject of active research. As we enter this new phase, let's analyze nutrition and gas exchange from different angles and evaluate its significance. The placenta intermediates the transfer of nutrients between mother and fetus. The perfusion of the intervolute spaces of the placenta with maternal blood allows the transfer of nutrients and oxygen from the mother to the fetus and the transfer of waste products and carbon dioxide back from the fetus to the maternal blood. Nutrient transfer to the fetus can occur via both active and passive transport. Placental nutrient metabolism was found to play a key role in limiting the transfer of some nutrients. Adverse pregnancy situations, such as those involving maternal diabetes or obesity, can increase or decrease levels of nutrient transporters in the placenta, potentially resulting in overgrowth or restricted growth of the fetus. Get ready for an enlightening exploration as we dig into excretion and understand its role in the broader context. Waste products excreted from the fetus such as urea, uric acid, and creatinine are transferred to the maternal blood by diffusion across the placenta. Welcome to the next segment, where we explore immunity and its significance in our journey. The placenta functions as a selective barrier between maternal and fetal cells, preventing maternal blood, proteins, and microbes, including bacteria and most viruses, from crossing the maternal-fetal barrier. Deterioration in placental functioning, referred to as placental insufficiency, may be related to mother-to-child transmission of some infectious diseases. A very small number of viruses, including rubella virus, Zika virus, and cytomegalovirus (CMV), can travel across the placental barrier, generally taking advantage of conditions at certain gestational periods as the placenta develops. CMV and Zika travel from the maternal bloodstream via placental cells to the fetal bloodstream, beginning as early as 13 weeks of gestation and increasing linearly, with the largest transfer occurring in the third trimester. IgG antibodies can pass through the human placenta, providing protection to the fetus in utero. This passive immunity lingers for several months after birth, providing the newborn with a carbon copy of the mother's long-term humoral immunity to see the infant through the crucial first months of extrauterine life. Antibodies, because of their larger size, cannot cross the placenta, one reason why infections acquired during pregnancy can be particularly hazardous for the fetus. Now, let's dig deeper into endocrine function and unveil the hidden treasures it holds within. The first hormone released by the placenta is called the human chorionic gonadotropin hormone. This is responsible for stopping the process at the end of menses when the corpus luteum ceases activity and atrophies. If it did not interrupt this process, it would lead to spontaneous abortion of the fetus. The corpus luteum also produces and releases progesterone and estrogen, and HIG stimulates it to increase the amount that it releases. HIG is the indicator of pregnancy that pregnancy tests look for. These tests will work when menses has not occurred or after implantation has happened on days 7 to 10. HIG may also have an anti-antibody effect, protecting it from being rejected by the mother's body. HIG also assists the male fetus by stimulating the testes to produce testosterone, which is the hormone needed to allow the sex organs of the male to grow. Progesterone helps the embryo implant by assisting passage through the fallopian tubes. It also affects the fallopian tubes and the uterus by stimulating an increase in secretions necessary for fetal nutrition. Progesterone, like HIG, is necessary to prevent spontaneous abortion because it prevents contractions of the uterus and is necessary for implantation. Estrogen is a crucial hormone in the process of proliferation. This involves the enlargement of the breasts and uterus, allowing for growth of the fetus and production of milk. Estrogen is also responsible for increased blood supply towards the end of pregnancy through vasodilation. The levels of estrogen during pregnancy can increase so that they are 30 times what a non-pregnant woman mid-cycle's estrogen level would be. Human placental lactogen is a hormone used in pregnancy to develop fetal metabolism and general growth and development. Human placental lactogen works with growth hormone to stimulate insulin-like growth factor production and regulating intermediary metabolism. In the fetus, it acts on lactogenic receptors to modulate embryonic development, metabolism, and stimulate production of IGF, insulin, surfactant, and adrenocortical hormones. Values increase with multiple pregnancies, intact molar pregnancy, diabetes, and ring compatibility. They are decreased with toxemia, choriocarcinoma, and placental insufficiency. In the upcoming section, we'll be shining a light on immunological barrier. The placenta and fetus may be regarded as a foreign body inside the mother and must be protected from the normal immune response of the mother that would cause it to be rejected. The placenta and fetus are thus treated as sites of immune privilege, with immune tolerance. For this purpose, the placenta uses several mechanisms it secretes neurokinins containing phosphocholine molecules. This is the same mechanism used by parasitic nematodes to avoid detection by the immune system of their host. There is presence of small lymphocytic suppressor cells in the fetus that inhibit maternal cytotoxic TMT cells by inhibiting the response to interleukins. However, the placental barrier is not the sole means of evading the immune system, as foreign fetal cells also persist in the maternal circulation on the other side of the placental barrier. Our focus now turns to DNA methylation, an important aspect of our discussion. The trophoblast is the outer layer of cells of the blastocyst Day 9 in figure above, showing the initial stages of human embryogenesis. Placental trophoblast cells have a unique genome-wide DNA methylation pattern determined by de novo methyltransferase during embryogenesis. This methylation pattern is principally required to regulate placental development and function, which in turn is critical for embryo survival. As we progress, let's shine a spotlight on other and examine its intricate interplay within our topic. The placenta also provides a reservoir of blood for the fetus, delivering blood to it in case of hypotension and vice versa, comparable to a capacitor. Now, let's redirect our focus towards clinical significance and discover its significance in our narrative. Numerous pathologies can affect the placenta accreta when the placenta implants too deeply, all the way to the actual muscle of uterine wall without penetrating it. Placenta previa, when the placement of the placenta is too close to or blocks the cervix. Placental abruption, premature detachment of the placenta. Placentitis, inflammation of the placenta, such as by TORCH infections. Now, let's shift our perspective and explore society and culture through a fresh lens, unlocking new perspectives. The placenta often plays an important role in various cultures, with many societies conducting rituals regarding its disposal. In the Western world, the placenta is most often incinerated. Some cultures bury the placenta for various reasons. The Maori of New Zealand traditionally buried the placenta from a newborn child to emphasize the relationship between humans and the earth. Likewise, the Navajo buried the placenta and umbilical cord at a specially chosen site, particularly if the baby dies during birth. In Cambodia and Costa Rica, burial of the placenta is believed to protect and ensure the health of the baby and the mother. If a mother dies in childbirth, the Aymara of Bolivia bury the placenta in a secret place so that the mother's spirit will not return to claim her baby's life. The placenta is believed by some communities to have power over the lives of the baby or its parents. The Kwajalein of British Columbia bury girls' placentas to give the girls skill in digging clams, and expose boys' placentas to ravens to encourage future prophetic visions. In Turkey, the proper disposal of the placenta and umbilical cord is believed to promote devoutness in the child later in life. In Transylvania and Japan, interaction with a disposed placenta is thought to influence the parent's future fertility. Several cultures believe the placenta to be or have been alive, often a relative of the baby. Nepalese think of the placenta as a friend of the baby. The Orang Asli and Malay populations in Malay Peninsula regard it as the baby's older sibling. Native Hawaiians believe that the placenta is a part of the baby, and traditionally plant it with a tree that can then grow alongside the child. Various cultures in Indonesia, such as Javanese and Malay, believe that the placenta has a spirit and needs to be buried outside the family house. Some Malays would bury the baby's placenta with a pencil if it is a boy or a needle and thread if it is a girl. In some cultures, the placenta is eaten, a practice known as placentophagy. In some Eastern cultures, such as China, the dried placenta zehek, literally purple river car, is thought to be a healthful restorative and is sometimes used in preparations of traditional Chinese medicine and various health products. The practice of human placentophagy has become a more recent trend in Western cultures and is not without controversy. Its practice being considered cannibalism is debated. Some cultures have alternative uses for placenta that include the manufacturing of cosmetics, pharmaceuticals, and food. As we enter this new phase, let's uncover the impact of additional images on our broader topic. Human placenta baby side-op human placenta immediately post-birth. Far-grey fetus of about 8 weeks, enclosed in the amnion. Magnified a little over 2 diameters. Far placenta with fetal membrane. Subplacenta with attached fetal membranes, ruptured at the margin at the left in the image. 5 with placentatis micrograph of a placental infection. CMV placentatis. 5 with placentatis micrograph of CMV placentatis. 5 placenta vasculature. Power Doppler. A Gifford power Doppler image of vasculature in 20-week placenta file. The placenta schematic view of the placenta. Human placenta maternal side of a whole human placenta. Just after birth. Human placenta fetal side of same placenta. Fetal side close-up of freshly delivered placenta. A close-up of umbilical attachment to fetal side of freshly delivered placenta. Placenta weight by gestational age. Placenta weight by gestational age. Zehek, dried human placenta used in traditional Chinese medicine. I love interacting with my viewers, so leave me a comment and I'll do my best to respond.